Okay, welcome everybody to the second webinar of the webinar series for the Rare Disease Cures Accelerator Data and Analytics Platform, an effort jointly coordinated by c-path and NORD under the auspices of the FDA. Today, we have a treat for you, the presentation by Jagdeep Adichetti, on data interoperability and artificial intelligence using a specific case study in polycystic kidney disease and kidney transplantation. RDCA-DAP launched, as you see on the slide, in 2019, and one of the main components of the effort is to set up the platform that can leverage data from multiple sources and use the data as the foundation for actionable solutions that can accelerate drug development in rare and orphan conditions. The platform is expected to be opened up and launched in September of this year. So this is in preparation for that big launch. A couple of housekeeping items. Please do keep your lines muted for the duration of the webinar. And at any time point during the presentation or during the panel session that is going to follow the presentation, if you have questions or comments, put them in the Q&A box, not the chat box. That's an option that you'll see on your right. One is called chat. Avoid that one. One is called Q&A. Do use that one for the questions. Please do address your questions using the function of the Q&A that addresses the questions to all panelists. But if you want your question to be addressed to a specific panelist, please indicate that in the question. Please avoid sending questions to the individual panelists, because that's a recipe for those questions falling through the cracks and not being addressed. And so with that, I'll introduce the speaker. Let's go to the next slide. So JD is a pretty remarkable individual. He's one of the few people on the planet that combine expertise in both pharmacometrics and artificial intelligence in a single brain. I've been lucky enough to supervise him during his initial tenure with c-path. He was also instrumental in the setup of the quantitative medicine program, and now he leads the predictive analytics group within the quantitative medicine program. He is definitely an expert in the integration of data and the transformation of data into actionable solutions, which is the main point and objective of RDCA-DAP. So with that, I'll leave you in the capable hands of JD to drive the presentation. One more thing, at the end of the presentation, you'll have a panel, and in the panel, we'll have Jeff Barrett, with whom I go way back. We've also been lucky enough to have him join the c-path team, and he recently took the reins of RDCA-DAP. We have Patrick Lang, who's a rising star within the team, another one of those big brains in artificial intelligence. And we have Alex, our scientific director for RDCA-DAP, who brings in a ton of both drug development and clinical expertise in orphan and rare indications. So with that, JD, take it away.
Thank you, Klaus, for the introduction and introducing the panel. So we'll start with the outline for the webinar today. First, we'll look at the hypothesis that we tested after we integrated the data sets. We'll look at harmonization methodology and modeling considerations. We'll look at data curation and visualization. We'll go over the AI ML methods for hypothesis testing, and then finally look at the conclusion. So for the hypothesis testing, we selected the following hypothesis. In PKD patients who developed ESRD and received a kidney transplant, the eGFR value for post-transplant can be predicted using pre-transplant data, including longitudinal eGFR and patient characteristics at the time of ESRD. So basically, our goal was to use the data from the pre-transplant PKD patient and predict the post-transplant eGFR value. And the way we did that was through analytics, looking at two different machine learning models. The first one was a long short-term memory time series model. So this is a more recent sophisticated model to typical neural network model. And here we accounted for longitudinal eGFR values and the patient characteristics at the time of ESRD to predict the eGFR value around 4 months post-transplant. We selected 4 months, and I'll go over in more detail why we did that, but the short answer is that was where we found most of our eGFR information for folks post-transplant. For our second model, we chose a relatively simpler neural net model with a few hidden layers, and I'll go in more details for that. And here, instead of looking at longitudinal eGFR values, we looked at the slope of eGFR for the PKD data and patient characteristics at the time of ESRD to predict eGFR value around 4 months post-transplant. Next, for our data sources, we got data from 2 different sources. The first one was PKD patient registry coming from Mayo Clinic. And this was acquired by c-path's PKD Outcomes Consortia. And the second data source was kidney transplant patient registry from Mayo Clinic, and this was acquired by the c-path's Transplant Therapeutics Consortia. And these data sources provide examples of what you would see on the RDCA-DAP platform. And the prototype that I'm highlighting today is gives you an idea of what sort of data integration and analytics and hypothesis testing that can be performed within the platform. For harmonization of pre-transplant and post-transplant data, we integrated these 2 data sources together by looking at the transplant date and gender. For the modeling consideration, we found that there were multiple eGFR values on the same day. So for that, we used an average eGFR values to avoid complications, to simplify it. So that's sort of one assumption that we made. And for post-transplant eGFR value prediction, we selected 3.7 months with 28-day window for the prediction. And I'll go in more details why we did a 3.7 plus or minus 28 days in subsequent slides. So looking at the disease continuum between pre-transplant and post-transplant, what you would see with the eGFR is that it reduces as you approach end-stage renal disease, and after which patients get a transplant, and the eGFR values recover with an eventual decline. And the shaded region that you see here is the analysis, the current analysis or the data that we have looked at. And you'll see this sort of profile when we integrate the data and visualize the data. So just to keep that in mind. For data harmonization, this funnel diagram gives a visualization of what we did with the two data bases. On the right-hand side, we have the PKD data. There we had 750 subjects. And on the kidney transplant side, we had 1,600 subjects. We looked at ESRD and transplant, so individuals that had an ESRD and got a transplant, we extracted them out. So there were 127 subjects, and then harmonized those individuals based on their transplant date and gender, which ultimately led us to 68 subjects, which we used to perform our hypothesis testing and analytics. So before performing the hypothesis testing and analytics, we looked at the summary stats, starting with the PKD data. Here's the summary stat for the longitudinal data. We had eGFR and serum albumin for the 68 individuals that we integrated. We looked at the mean number of time points that these subjects had, and then looked at some other metrics as well. And the purpose of this was to get a better understanding of the data that we had harmonized, and then come up with the hypothesis that we want to test, so which I already mentioned. But in order to get to that hypothesis, get to the type of analysis we want to perform, we sort of had to look at what data we are working with. And that's sort of the exercise we do in terms of looking at the summary stats distribution of these variables. So the other longitudinal data that we had was the height, weight, BMI, blood pressure values. We also looked at how much information we have, the richness of this information that would help us figure out what sort of hypothesis do we want to test. And then in addition to the longitudinal information, we also had other background cross-sectional information for the individuals. So here you would see a list of those. I won't go into too much detail, but the idea is apart from the longitudinal information of eGFR, in order for us to build a model that would have good predictive power, we would want to also include in other information that could help with the predictability, right? So these were the other variables that we considered for the analysis. And we'll come back to that, to what specific variables that we used for the analytics in the subsequent slide. Now moving on to the post-transplant. So far we looked at the pre-transplant in the PKD side. On the post-transplant of things, we did something similar. So here the longitudinal variable we had was eGFR, and we looked at the mean number of time points, range, and so forth. We also visualized the trend in eGFR. So the visualization, you could see two of them here. The first one on the left-hand side is all the data we have. And you can see it's clear that majority of the information we have is within the first year or year and a half. And then on the right-hand side, you would see the timeline, the time from transplant. We just looked at the first 1.5 years to see what that trend line looks like. The blue line you see here is the lowest line, and that's typically used to just look at the trend, a general trend in the data. We won't focus on that for this effort, but our purpose here was to figure out what is the richness of eGFR value, and then what sort of hypothesis testing we can perform with the data we have on the post-transplant side of things. So in that vein, we looked at the frequency of visits or eGFR values we have in the post-transplant data. And here you would see on the left-hand side, we have majority of the eGFR values around baseline or time of transplant, around 2 to 4 months, and then 1 year. Within the first 1.5 years, which is where majority of the data is, as I mentioned in the previous slide. And we also looked at what does this data look like, what is the frequency of eGFR values in the whole data set. So for from time of transplant to 6 years, and you could see after 1.5 years, it's pretty sparse. So then the next step was, okay, within the 1.5 years, what is the time point where we can maximize the number of individuals that have the eGFR values? So we, as you can see here, we are going towards finding eGFR a time point where we have the maximum number of individuals with eGFR value, right? So that the total number is 68. That's the number of individuals we were able to integrate between the two databases. So what we did was, since we see that the eGFR values are quite frequent, around 3 to 4 months, we looked at these eGFR values with varying windows, right? So here on the left table, you'll see different time windows, 7, 14, and so on, and then at different time points, 3, 3.5 months, 4, and 1 year. And then within those time points, we found that 3.5 to 4 months is the sweet spot. The other consideration is also how far you want to go in terms of predicting an eGFR value. So since the purpose here was to use the PKD data or pre-transplant data to predict an eGFR value in post-transplant, the further out you go into the post-transplant data, the harder it becomes for you to predict. And you can imagine, because as you move further out from the transplant, there will be more variability that you have to capture, and you're not really using the data on the post-transplant side to make that prediction, right? So you're using the data on pre-transplant side. So that was another consideration why we selected 3.5 to 4 months. But in any case, so within 3.5 months and 4 months, we then zoomed in. So here on the left, on the right-hand side, the table, you'll see that we looked at 3.5, 3.6, and so forth, till 4 months. And then what we found was at 3.7 months with a window of 28 days, we can capture 63 subjects. So that's sort of what we selected as our outcomes. So basically, trying to predict the eGFR value at 3.7 months, plus or minus a window of 28. With the window, you want to make sure that it's not too wide. So that there's a level of judgment you have to make there. But in any case, so that's the outcome that we looked at. But again, there are other outcomes that you can look at, or different hypotheses that you can test. So once we integrated the data and plotted the integrated data for those 68 individuals, this is what it looks like. So this graph involves information from the pre-transplant, which is on the left side of the 0, which is the time of transplant. So this region here is your PKD region, and then after 0 time point is your post-transplant. And you can see the trend line, the lowest line, blue line, sort of resembles to what I was talking previously in terms of looking at the continuum and the analysis space that we are looking at, right? So it sort of aligns with that. So I want to change the gears a little bit and provide a little bit of background around the type of methodologies we used, which was in AI/ML. And the reason I want to do that is it would help you get a little bit more, a better understanding of why or what the model looks like and what it does, right? So that's sort of the purpose here, is to provide a little bit of background to see what part of the AI/ML landscape did we select our models from. So the AI landscape in general is pretty broad, and here I try to represent that in two sub categories. The first one is machine learning, the other one is natural language processing. I'm not going to go into too much detail around natural language processing. A lot of you might be familiar, but we leverage the machine learning side of things, more specifically deep learning side of things. And with the deep learning, there is neural networks. So we used different architectures of neural networks in order to do our analytics. and go in more details of which one, what was the architecture. So within the neural network, this is an example of a typical neural network, or a very simple neural network, just for our understanding. And it consists at a high level of 3 parts. You have the input layer, you have hidden layer, and output layer. And you can create different versions of the neural network based on your requirement. And when it comes to deep learning, it's essentially a neural network, but has multiple hidden layers. So here you would see that we have 1, 2, 3, 4 hidden layers and an output layer. So technically, this would be called a fully connected 5-layered deep net. And typically, the input layer is not considered as a layer. And the reason it's fully connected is because if you look at any neuron in the hidden layer, it's connected to every other neuron or input in the previous layer. And the information flows from left to right. And the way you optimize it is through different methodologies, and one of which is a gradient descent, but we won't go into that detail. But I do want to spend a little bit of time on the building blocks, because that would help us understand the architecture of the specific methodology that we are going to be using for this effort. So if you look at a particular neuron, artificial neuron within the neural net, it would sort of resemble the biological neuron. So where you have the nucleus and you have information coming through the dendrite into the nucleus, and it goes through an activation, and information passes through the axon to the next neuron. Similarly, in a artificial neuron, you have information coming from say the inputs, and it's connected to the artificial neuron, and each of these connections have a certain weight, which determines the magnitude of or the strength of that connection, and that is summed up and added to a bias terminology, and I'll go into a little bit of detail there, and goes through an activation function to give you an output, and this output would be fed into the subsequent neuron. So what are the components of a neural net? There are 4 basic components. There's the input, weight, bias, and activation function. And the bias is essentially an offset for the sum of weights, so that to ensure that the sum crosses a certain threshold. The activation function basically determines if the neuron will fire or not. Additionally, it provides ability for the neural network to capture nonlinearity within the data. So then, looking at the specific equations that we used to calculate the output from a single neuron. So the y hat is your output, which is basically the sum of the weight and input, which is given by this, added to a bias. And so this is the linear portion. And then you add an activation function to it to get you the output. And you do this for each neuron in your network. So you can imagine a more complicated neural net. And these are the building blocks for that, for any neural net that you would build. The way we compute these input and the output is to create an input matrix and a weight matrix. And typically in most softwares, you would do a matrix operation, which is typically a dot product between the two. Not going to go into too much detail. This just provides the background of how we approached the analytics. So now coming to recurrent neuron. So far, we looked at a neural net that you can input a cross-sectional information, right? In our case, we had longitudinal eGFR information, and we want to be able to input that longitudinal information within the neural net and be able to predict the eGFR value on the post-transplant side of things. So in that case, instead of a typical neuron, you have something called a recurrent neuron, which is essentially a normal neuron, but has the time component to it. So if you look at the neurons that are shown in red, when you unfold, or if you look at the longitudinality of that neuron, you would see that it captures the time component as well. So the YT-1, YT, and YT+1 are the outputs of that particular neuron at different time points, so T, T-1, and T+1. And the way you calculate the Y of T is using this equation, which is very similar to the one I just showed. The only thing is that you would incorporate the output from the previous time step. So to calculate YT, you would use YT-1 times the weight of YH, right? And that allows you to perform or input longitudinal data and be able to make predictions. Now, there are some disadvantages of the recurrent neural network, or RNN. The biggest disadvantage of RNN is that if you have a long time series, it tends to lose information from the earlier time points, right? And one fix to that is to use a long short-term memory neural network, which is shown here on the slide, the visualization of how the network looks like. I won't go into too much detail on each of the components, but I do want to highlight the important parts here, which is the short-term and the long-term portion. So in order to account or keep the information, when you have a longer time series, you would be able to use the long-term part of the Lstm to ensure that you are not losing information. And then the short-term is make sure that you can capture the longitudinality in the vicinity of a time point. So this is what we used for our hypothesis testing as one of the machine learning models. And I'll show you in more detail what that architecture looks like. So for our workflow, we followed a typical workflow that is used for machine learning. Once we have our data that we integrated, we assemble the data set. We split the data randomly into training set and validation set. We did it in 80-20, but you could do it 90-10 or 70-30, depending on your use case. A lot of times people have millions of samples. In that case, you don't really have to do 80-20. You could just leave a few thousand aside for validation. Once we have our training set, we perform model training. So for our purpose, we used two models. One was the Lstm, the other one was a neural net model. And once we have the model trained, we would perform model validation to look at how well our model did. And for our performance metrics, since we were doing a regression problem, we used mean squared error, MSE, and mean absolute error. So essentially here, you're trying to minimize the difference between the actual and the predicted value. There are other metrics as well that are used. For our purpose here, we used mean squared error and mean absolute error, and optimized to ensure that these two are minimized. Before performing the analysis, we did a quick visualization of the data set we're going to work with for the analysis. So here you'll see this is similar to what I showed when we integrated the data, but here the data is a bit more limited to analysis. Here you see the 0 is the time from ESRD or transplant. They were very close to each other. You'll see the profile of eGFR in the pre-transplant, and then we have our eGFR values at around 4 months, right? We also looked at some other percentages of wide percentage male and so forth, just to get an idea of what our data looks like. For our first model, which was the Lstm model, we used the baseline variables here that I've listed. This comes from the summary stats that I was showing. I won't focus on that too much, but I do want to look at the longitudinal variable and provide information around what we did different than what you would see typically, right? So in our case, we had the pre-transplant EGFR data, but it was sparse. So the EGFR values for individuals were not at the same time point, and not every individual had the same number of EGFR values, right? So in that case, we wanted to make sure we use all the information. So what we did there was to create another longitudinal variable called delta, and this delta captures the time between subsequent measurements. So essentially, we want to tell the model that, yes, the individuals don't have EGFR values at the same time point, but here's information of what is the difference between the two time points, and provide that information as well to the model so that it can properly train. And then, of course, the outcome is our eGFR post-transplant. So now looking at the model architecture, here's what the Lstm model architecture looks like. I just want to highlight the important portions of these. The way we approached it is we had the 37 time points eGFR for individuals, and we use that for our Lstm model training. So you would see that that goes into the Lstm layers, and we used a dropout layer for regularization to ensure that there's not much, there's little overfitting, right? And then the remainder of the information, the background information, the cross-section information, we use that after the Lstm layer to concatenated that with the output we get from the Lstm layer, which goes into the, so those two goes into the dense layers, and we get the output, which is the predicted EGFR values around EGFR value around 4 months. These just gives you information of what we got, but essentially what we get as an output is the EGFR value of 4 months. And this one is, to be specific, this one is the information we get after the Lstm layer. So we just output that for informational purpose. So the total trainable parameters were 6,930. So these are majority of your biases and weights that I was showing you before. And so you can imagine that we need to optimize the values of these total trainable parameters through the data and optimization methodology. Okay. So for results, what we found was that the Lstm model performed quite well with training data, but showed potential overfit with the validation data. So here on the right side, you would see the blue plot is the training, and the mean square error is what we were calculating, and it goes down, which is what it's supposed to do as you continue model training. But we see that the red line, which is the validation, is not going down; it's actually going up, which is a normal. It indicates that there is a potential overfit. So with that, what we did, we then decided to use a much simpler model to see if we can overcome the overfitting issue that we were having. And then with that in mind, what we did was, instead of looking at longitudinal EGFR value, we calculated the slope of the EGFR value for the pre-transplant site, and also looked at the last EGFR value pre-transplant, so that we can provide that information as well, along with the slope to the model to be able to predict the post EGFR. And for that, this is the model architecture. As you can see, it's much simpler model architecture, and it sort of resembles to what I was just showing you in terms of a deep net architecture. So this is a fully connected neural network where we input the 13 variables as the input into a dense layer with 64 neurons and another dense layer with 32 neurons to give us the output, which is our eGFR value at 4 months, around 4 months. And then for here, the total trainable parameters were 3009. As you can see, it's almost half what we had for Lstm. Looking at the results for neural net, what we saw was that it performed relatively better than Lstm with the validation data. And you can see that here with the plot. When we look at the error, you would see that the orange is the training, which is going down. That's what we want to see. And then the blue is pretty much remaining flat, which shows us that there's not much overfit compared to the one we saw with Lstm, although the mean squared error was relatively high in this case. But we want to ensure that you are able to generalize the prediction of the model to unseen data. So that was the purpose there. So then we looked at the observed versus predicted for the validation set. So here you'll see that between model 1 and model 2, although we had limited data with the validation, we would see that model 2 is relatively better in terms of the prediction compared to Lstm. So in conclusion, for data interoperability side of things, we were successfully able to integrate data from two sources, PKD, patient registry, and kidney transplant patient registry. And for our analytics and hypothesis testing, we found that the model performed well during training, but it showed potential overfit, especially with the Lstm model. However, with additional data, we believe the eGFR prediction of 4 months post-transplant can be improved. And also, we can explore other research questions and create a different analysis set with additional data as well with the data we integrate or a different prototype as well. So finally, I would like to thank our data and analytics team. With data curation visualization, we had Emily Hartley, Eric Fry, Bob Stafford there to help us integrate the data. With predictive analytics, Patrick Lang and Megan Keller did the model analysis, and then Klaus and I provided technical supervision. So with that, I would hand it back to Klaus for our Q&A session.
Thanks, JD. That was certainly a very informative presentation. Okay, I'd like to invite the panelists to jump on board and turn on their videos and get ready for the panel discussion. All right, we do have a couple of opening questions from the audience. So thanks to the members of the audience for the questions. The first one, I'm just going to assume this is for JD, since it's a very valid technical question. Do the backward connections in a recurrent net, the neural network you described, go back only one layer as a rule, or not? And if not, why? Can you tackle that one, JD?
So I believe the question is around backward propagation. So that is what happens during the training. So in the forward propagation, we calculate the output from each of the neurons, which is the white hat that I was showing, and you get the prediction, right? And typically when you do the first pass, your prediction is way off to what the actual value is. So to optimize the model, what you would do is you do a back propagation. So essentially going backward and creating and calculating the gradient. And the idea is to use optimization methodologies like gradient descent to minimize that difference in your prediction versus your actual value. And then you do that multiple times. So you do a number of different iterations to minimize that difference between the actual and the predicted value.
Excellent. Thanks, JD. There's another question also from the audience pertaining about the fact if you also experimented with the structure of the network, for example, the number of layers, nodes per layer, and other hyperparameters when you build the models.
Yes. Yeah. So we looked at a few different things, right? So in order to improve the performance, you can optimize the hyperparameters, and one of the hyperparameters is the number of neurons, number of layers, and so forth. So we did look at a number of different things. As you can see, we looked at two completely different architecture as well, right? And within each of these architecture, we looked at different variations of these architectures. So different variations of Lstm and different variations of neural net. But at the end of the day, I mean, there's only so much you could do to improve performance with your model, right? And then there's a portion of how your model performs is dependent on the type of data, richness of the data, and the amount of data you have, and the amount of signal you have within the data. So.
Excellent. Thank you, JD. Patrick, do you want to add anything to that?
Yeah, I just wanted to jump in on the first question about the backward connections in the recurrent neural network. In the case that it wasn't about back propagation, the way I interpreted that question was, does the recurrent neural network, does each unit look back just one time step? And the yes and no, it receives the long-term kind of dependency from the previous time step, but in theory, the information is retained from all previous time steps. So while it does look back just one time step, it should be able to pick up information from the time steps that preceded that.
Excellent. Okay, moving on to another question. This one pertains to any plans for additional prototypes similar to this. And JD, while you answer that, I would also ask you to emphasize what this exercise provides as a proof of concept for RDCA-DAP as a platform beyond what you have shown for the interoperability between PKD and kidney transplantation data.
Right. So yeah, that's a good question. So as I was mentioning during my talk, the prototype that we showcased here gives you an example of what capabilities you can expect from RDCA-DAP, right? And what sort of data integration methodologies you could use, and what sort of analytics you can perform, hypothesis testing you can perform. Now coming to the question of what other prototypes we could look at, there are other examples in inborn error of metabolism diseases or lysosome storage diseases. And Klaus, you can mention, you might have other ideas there as well that you can mention, but so there are multiple other areas that we can focus on in terms of data interoperability and doing hypothesis generation, showcasing the ability of the platform to do so.
Excellent. Jeff, following up on that topic, can you elaborate on why these kinds of hypothesis generation solutions are of added value in drug development, especially for rare and orphan diseases?
Yeah, it's a great question, Klaus. I think we all appreciate in the rare disease setting that there's certainly gaps in knowledge, and we have to be flexible in terms of both the approaches that we take to filling in the gaps, as well as use this as a way to solicit additional data, and that could come from real-world data sources as well as data from clinical trials. So I think the promise of the future development of RDCA-DAP is this home for collaboration that incorporates both data and tools. I think JD's examples gives you just a fantastic look at what's possible using more sophisticated methodologies when we don't always have the most refined data sets, and then learning to make the most out of the data that we have, in addition to providing guidance for where data could be collected better and improving the quality is really the intention of the overall effort. Thanks, Klaus. Good question.
Excellent point. And that actually is a good segue into another question from the audience. And perhaps I'll ask Alex to address at least the one of the main components of this. The question pertains to the actual methods, right? So deep learning algorithms and how that those kinds of approaches are data intensive, and how that relates to the fact that one of the realities of rare and orphan conditions is that data sets are limited. So Alex, can you help us illustrate what the big vision is for RDCA-DAP to be able to integrate multiple data sources, in addition to what Jeff alluded to already in the written response?
Yes, thanks, Klaus. So on a very broad level, as everybody's probably aware, one of the issues that drug development is facing when dealing with rare disease drug development is the paucity of patients. Rare disease can be from ultra rare to 100, 200, and 1,000 patients in the US. So potentially you're facing disease with a very limited number of patients. And so the studies don't have a lot of patients in them. And so there's limited knowledge that you can extract from one trial with maybe 20 patients. So one of the big goals of the platform is to be able to aggregate as much data as possible from different sources, so clinical trials, but also patient registries and natural history studies, and for the future, more types of data, such as electronic health records. So the idea is to provide a place where you gather all this data and provide an avenue for leveraging as much knowledge as possible from these aggregated data sets that we have within the platform. So this is one of the main goals for RDCA-DAP, is to create a place where scientists can access all this data, but be able to integrate this data and increase the knowledge that they can extract from analyzing these integrated data sets. And so I guess on the more technical level, I'll let you or Jeff give more explanations.
Yeah, exactly. So I'll just comment in addition to that the intention of the exercise, as JD illustrated, was as a proof of concept to determine if we could take two examples of two disparate indications, both with an orphan status designation, and if we could make the data interoperable to run these kinds of explorations. So the intention of the exercise was, even though it provided some meaningful answers in itself, the intention was not so much about the answers that the exercise could provide in terms of the eGFR, longitudinal behavior, et cetera, but more around the feasibility of being able to make the data interoperable across conditions. And I think that's a good segue into a couple of questions from the audience that relate to two sides of the same coin, and that has to do with the data. So the first part of the question from the audience has to do with how you were able to identify the individuals that may have participated in one aspect of the data set, and then in the other aspect of the data set once they cross this threshold of end-stage renal disease and got a transplant. So JD, I'll let you go first with that one, and then we'll switch to the other side of the coin of that question, which is on an important one about privacy.
Mm-hmm. Yeah, so I think you are touching upon the fundamental question of data interoperability, right? So in our case, when we looked at the PKD data and post-transplant data, although they come from Mayo Clinic, they do not have the same subject ID. And with the data set that we have, we were working with, the way we integrated them was to look at the transplant dates in the two data sets and look at the gender. That was the way we integrated the two data sets. That's not the only way to integrate it, right? Depending on the data sets you have or data sources you're getting it from, you could find other ways to integrate it. A lot of times, in reality, there might not be, it will not be feasible to identify subjects in two data sets, right? And you might have to find other ways to integrate it. And there are implications of how you integrate the data, and you would see those implications in time when you go about hypothesis testing or trying to predict or create a predictive model from one continuum to the other continuum, right? So if you are able to integrate the data very well, like robustly, you would see that your predictive performance is much better. But if you are making a few assumptions and there are some limitations in terms of how you're integrating the data, then you will see that you will lose some information as you go from one continuum to the other continuum. And we just showed one of the examples, like you were saying, Klaus. The purpose here wasn't to get the best model ever, but it was more of showcasing the capability of what is possible, right, within the platform.
Excellent. And that actually brings up a segue into the next point, which is the data privacy side of things. But let me just add a comment about what JD just mentioned. Even in a single disease, where you could have the integration of multiple data sources, as Alex was mentioning, registries, observational studies, and if there are those data available, randomized clinical trials. In rare diseases, it could be a reality that you have patients and individuals participating in multiple studies. And then the question is, what happens if you can't reliably pick out the individuals that participated in multiple studies? What is the actual consequence? Well, in reality, it's an artificial shrinkage of the residual variability. And if you recognize that up front, that's a limitation, that you have that potential, there are ways of dealing with that caveat, especially when you're presenting these kinds of solutions in a regulatory environment. But let me switch to the other part of the question. I'll probably ask Jeff to start providing the answer regarding how the platform in general protects the identity of the subjects, because data privacy is, of course, a major concern. And one of the things that we put up front at any data integration effort at c-path, but in the case of rare diseases, that's an even more important consideration. So Jeff, can you provide an overview of how the platform protects the privacy of the individuals?
So before I get into that, what really distinguishes RDCA-DAP from other rare disease platforms is the who and the how. On the who side, this platform was built for the entire rare disease community, but first and foremost is the patients themselves. The patients are in control of their own data. They have the rights at any time to rescind their data from incorporation into the platform. And that's true from any point that we get the data from. So I think we're very much cognizant of the who and the patients being in control of their data. But it's also built for pharma partners, for stakeholders in this area, for reviewers at regulatory authorities around the world, and for foundations and other people who will also facilitate the provision of the rare disease data. What Alex and others have brought up is the paucity of this data. So we have to make the most of a very limited and precious data source. Now, the how side is the detail associated with how do we do this with confidence, securely, and with patient privacy in mind. And that's really dictated by adopting FAIR principles. Now, that FAIR in that concept is not just a nice word here. It's a complete set of best practices that allows us to transfer only de-identified data, to maintain it in that form, but also behind the scenes to keep a lot of the global identifiers in place so that we can integrate data with confidence, that we don't get into concerns around duplicate data, but again, without identifying the data in any means. This is a complete work in progress. We are adopting very futuristic methodologies and approaches to accomplish this. This is something that we learn from with partnerships around the world, and with conversations around the Global Data Privacy Act. So there's a GDPR compliance component of this, and it's something where we have a lot of complicated heuristics. The platform keeps track of the sharing conditions as well as the access piece of this. So there's a full audit trail and a recognition that the metadata that we collect is there to help us facilitate the construction of of suitable data sets. And, we will use all of these methodologies and approaches. So more modern approaches such as GUIDs are behind the scenes that allow us to construct this in a very reliable manner. And it's something that we're also willing to modify and learn along the way. So a lot of the dirty work behind the scenes is part of the functionality of the platform.
Beautiful,
and we're
Jeff.
open to other suggestions too.
Exactly. This is great, and thanks to the audience for the engagement and the questions. Looking at the clock, I think we're gonna adjourn now, but let me close with the following comments. So first off, JD, thank you for the very illustrative presentation. Second, thank you to you, the panelists, for having this engaged conversation. But most importantly, thanks to the audience for putting in the questions, very relevant questions. I'll close with a call to action for everyone that attended the webinar. If you want to follow up with us and be part of the journey, you have the contact information. Please do reach out, and we would be more than happy to bring you up to speed as to where things are, and get you engaged and have you travel the journey with us. This is a very exciting effort, and as Jeff said, it's pretty unique in the way it has been set up, and it'll provide a good marriage of the push for data sharing, data interoperability, and open science with the all-important protections of privacy. So this is going to have ramifications where the rare disease and orphan disease field is going to provide valuable learnings for other diseases. So, like I said, very exciting. I'll thank you all for the time, and with that, I think we can adjourn the webinar. Thank you so much.
Thanks, JD. Good job.
Thanks, everybody.
Thank you.